The Rift Valley fever virus causes the highly pathogenic RNA virus, the Rift Valley fever. This zoonotic virus is prominent to the country of Africa. It is spread through the vectors of mosquitoes, resulting in high mortality rates up to 90% in ruminants, with an additional 100% abortion rates. There are approved emergency use animal vaccines for RVFV, but there are no FDA-approved vaccines for humans. Humans with RVF can experience symptoms of permanent vision loss due to retina inflammation and severe hemorrhaging, which seems pretty bad. But considering we haven't heard much about Rift Valley and it's been here for 90 years, it's probably not a big deal, right? But it's completely wrong. The only reason this virus has been contained and minimal thus far is because there have been little to no instances of person-to-person transmission. However, what if this were to change overnight? How could we contain an RNA virus so pathogenic? It would be another costly pandemic that could only be saved by vaccines. Extracellular vesicles are vesicles derived from eukaryotic cells that can potentially lead to vaccine candidates and therapeutics against Rift Valley fever. This is a conceptual model explaining how extracellular vesicles could potentially be used to induce an antiviral protective response in cells. Viral RNA from the Rift Valley genome can be inserted into extracellular vesicles to trigger a receptor called RIG-I. RIG-I stands for retinoic acid inducible gene 1. This is a receptor present in all eukaryotic cells within the cytoplasm that can detect Rift Valley RNA or any viral RNA. This RIG-I receptor can in hand induce an immune response through interferon beta activation, acting like an alarm of invasion, indicating the need for protection. Now, through this research project, we aim to understand the exact type of RNA responsible for this immune response in cells, because the RVFV genome is approximately 12 kilobytes with an immense amount of RNA present, but it's difficult to determine which RNA species in particular is responsible for this. In order to understand which RNA species is responsible, there are a set of methodologies that need to be conducted. First being infection, which is infection of cells with the Rift Valley virus. Transfection is inserting viral RNA into eukaryotic cells. RNA extraction involves isolating the RNA from cell contents, and real-time polymerase chain reaction will analyze the amount of interferon beta activation occurring in each sample. Out of these 5 steps, infection, transfection, RNA extraction, and polymerase chain reaction have been conducted successfully. The results here show that an ideal ratio of poly-IC, which is an RNA mimic that induces interferon beta activation, to a ratio of DOTAP, which is a transfection reagent, is ideal in the ratio of 10 to 30. Now, since polymerase chain reaction has confirmed the best use of a 10 to 30 ratio, the next step would be using Western blot in the same transfected samples with the same ratios to determine that a ratio of 10 to 30 to poly to DOTAP would work ideally as well.